Now let's spend some time discussing the genomic properties of viruses. Guys and girls, this is where the money is. The genetic material of a virus is the key characteristic of how we classify them. On step 1, it is very plausible for the board examiners to present a virus or present a disease clinically and then ask you to identify the virus based on the genetic material rather than the name of the virus. So let us start with the basics. Can someone tell me from biochemistry, what are the two types of nucleic acids? I hope we're all saying there's DNA and there's RNA. We can see from this image that RNA is single-stranded, there is just one strand of nucleic acids, while DNA is double-stranded. This mostly remains true in virology, except for a few exceptions which we will go over together. Let's start to discuss the different DNA viruses. There are 7 DNA viruses, and they are all happy. We can use happy as a mnemonic to remember that the 7 viruses include the hepatinovirus, the herpes virus, the adenoma, polyoma, papilloma, parvo, and pox virus. I know it's a mouthful. Hopefully this mnemonic happy helps you remember the 7 different DNA viruses. All the DNA viruses are double-stranded DNA, just like in our cells, except for one, and that's the parvovirus. We can remember that parvovirus is single-stranded by remembering the mnemonic parvovirus is part of a virus. All DNA-stranded viruses are icosahedral shaped. They have an icosahedral shaped protein capsid, except for one, which is the pox virus. The pox virus is the only DNA virus that is not icosahedral, and it has a complex bullet-shaped protein capsid. All of the DNA viruses replicate in the nucleus of our human cells. All of the DNA viruses replicate in the nucleus of our human cells, except for one, and that again is the pox virus. So the pox virus is different than all DNA viruses in the fact that it does not replicate in the nucleus and it is not icosahedral shaped in terms of its protein capsid. So now let's spend a moment to talk about DNA viral replication. Remember, the key concept in virology is that viruses rely entirely on other cells, human cells, animal cells, to replicate themselves. DNA viruses, as we mentioned, take advantage and replicate in the nucleus to use the DNA and RNA polymerases that are present there. They use the DNA polymerase to replicate their DNA, and they use the RNA polymerases to convert the DNA into the messenger RNA that's required to then be translated into protein to build the viral capsid. So now this brings us back to the central dogma of biochemistry and molecular biology. The DNA must be transcribed into messenger RNA, and where does this happen again? This happens in the nucleus, very good, with the RNA polymerases. Then, once mRNA is made, it leaves the nucleus and then is translated into protein. This process, as I mentioned, occurs outside of the nucleus by the ribosomes, either in the rough endoplasmic reticulum or in the cytoplasm. Very good. Bring back some cell biology here. Once the proteins have been made, they are then composed and brought together to form the virin. This process occurs for all DNA viruses except for one, and that's the pox virus. All DNA viruses replicate their DNA and produce their messenger RNA in the nucleus, except for the pox virus, which does all of this on its own out in the cytoplasm. So wait a minute. If the pox virus is doing all of this in the cytoplasm, what do you think the pox virus needs to be able to do this? Well, remember, if the virus doesn't go to the nucleus, it needs to find some way to replicate its DNA and transcribe its DNA to mRNA. There's no polymerases in the cytoplasm normally. So what that means is that the pox virus actually takes with it and carries with it its own DNA and RNA polymerases. So essentially what it does is it carries its own enzyme so it doesn't require the nucleus to replicate itself.